We will begin this session with Dr. Élyse Caron-Beaudoin. Dr. Élyse Caron-Beaudoin is an assistant professor in environmental health in the Department of Health and Society at the University of Toronto, Scarborough. Her research is focused on the development of transdisciplinary community-based research projects to assess the impacts of anthropogenic pressures on health by combining information from multiple levels of biological organization. Elise holds a PhD in biology with a specialization in toxicology from the Institut National de la Recherche Scientifique, Armand Frappier Institute in Laval, Quebec. From 2018 to 2020, she completed a Canadian Institutes of Health Research postdoctoral fellowship in the Department of Occupational and Environmental Health at the Université de Montréal. During her fellowship, Dr. Caron-Beaudoin investigated the associations between density and proximity to hydraulic fracturing wells and birth outcomes in Northeastern British Columbia. She is currently investigating gestational exposure to environmental contaminants associated with unconventional natural gas exploitation and their endocrine disrupting potential. She is a collaborator and co-investigator on several other research projects in environmental and Indigenous health, including in the Arctic. Please welcome Dr. Caron-Beaudoin.
First, I want to thank the organizing committee for the invitation today. I'm very happy to be with you and to present some of the work that I've been fortunate to be part of regarding unconventional natural gas and human health. Before I start, I first want to acknowledge that I am thankful to live, work, and play on the traditional territory of many First Nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, also known as Toronto. So today I will be speaking to you about some of the community-based research projects our lab has been implicated in regarding UNG or unconventional natural gas and health impacts in Northeastern British Columbia. And briefly, I just want to present to you our lab from bench to communities. So our lab highlights community knowledge as a valuable source of information in order to explore the relationship between environmental factors and human health. So we pursue the following objectives. First, we develop customized research strategies to identify potential environmental factors associated with health issues observed by community partners. We then investigate the prevalence and incidence of those health issues when possible. We also developed or use cellular bioassays to really study the mechanisms involved in those health issues. And we hope to contribute to adequate environmental and public health policies by collaborating with regulatory agencies. So I think everyone here is familiar with UNG. I don't have to go over that, but just to give you a bit of an idea of the landscape in Canada. So here you have a map of Canada, and I'm going to talk today about Northeastern British Columbia. So British Columbia is at the very west part of the country, and our research is being conducted in the northeastern part here of that province. This is a close-up of the area that you can see here. So in Canada, around 25% of natural gas production is done in that region using mostly hydraulic fracturing. You can appreciate the extent of the development on the map here, the close-up, where all the little colored dots represent oil and gas wells or facilities. This particular region sits on the Mutney Rock Formation, which represents approximately 67% of British Columbia's production of natural gas. And we know from previous scientific literature that there is potential contamination associated with that industry, namely by volatile organic compounds, or VOCs for short, and also trace and radioactive elements that are naturally occurring in those rock formation. So I want to speak a bit about the toxicological evidence in terms of potential mechanisms of toxicity associated with chemicals associated with UNG. So in 2016, Elliott and colleagues published a very interesting study. They did a systematic evaluation of around 1,000 chemicals that have been identified in hydraulic fracturing fluids, and they screened those chemicals for their potential reproductive and developmental toxicity. So they gathered all evidence from cellular model studies, animal models, and also human studies. What they found is that for around 76% of the identified chemicals, information on reproductive and developmental toxicity was lacking. On the remaining 24% of those chemicals for which they did have toxicological data, 43% of them were identified as developmental toxicants, 40% of them as reproductive toxicants, and 17% of them as both. It's important to note that when Elliott and colleagues did that evaluation, there was no peer-reviewed exposure assessment studies in the context of UNG in the scientific literature, meaning no published levels of exposure in local communities. However, there's a multitude of communities' observations in Northeastern British Columbia regarding the impacts of UNG. Indeed, people relying on traditional or wild foods and or spending time on the land are aware of potential contamination and condition of their environment. Local communities' knowledge, experience, and observations can be used in conjunction with biomonitoring, toxicological, and epidemiological data. That is, that communities' knowledge is an epistemology worthy of respect that stands on its own. And as such, we believe that environmental health projects should seek and target observations and concern that communities have specifically flagged. So in 2012, in Northeast BC, a number of First Nations reported concerns about consumption of elk, moose, and deer, which are a major food source in the communities. Specifically, they observed noticeable declines in those populations, and hunters also reported several abnormalities in the animals, such as tumors and growth. And their observation, they thought, were caused by potential exposure to contaminants from increased oil and gas activities in the region over the last decades. They were invited to provide input on potential health risk and areas of concern to the Fraser Basin Council, but they said that the timeline to partake in consultation was too short, and that led to no validation or analysis of the concerns provided by communities, and rather those observations were presented as anecdotal events. So it is in that context that our studies were developed following discussion with the Treaty Tribal Association, the West Moberly and Saulteaux First Nations, as well as local healthcare providers. And one of the main areas of concerns we identified was regarding exposure assessment. So the available toxicological data that I presented earlier tells us that reproduction and fetal development might be affected by exposure to UNG chemicals. And because human health risk depends on both the toxicity of those chemicals and also our level of exposure to those chemicals, we were first interested in conducting an exposure assessment pilot study in pregnant women living in Northeastern British Columbia. So this pilot study was conducted in 2016. The principal investigator was Marc-André Bernard at Université de Montréal. We received funding from our institution and the West Moberly First Nation. We worked with 2 medical clinics and a quite diverse research team. It was a community-based research project. So the Treaty Tribal Association, West Moberly, and Saulteaux First Nations were partners in that pilot study. Very briefly, to go over some key findings of that initial pilot study. So we measured a variety of volatile organic compounds metabolites in urine samples of those participants, and we also measured trace elements or trace metals in urine and hair samples of the participants. And we compared what we measured with the general Canadian population. So we found that the level of one benzene metabolite, benzene is a VOC, so this metabolite is called trans-transmeconic acid, or TTMA. So the levels in the participants of the pilot study were quite higher compared to the general population. And they were even higher when we looked only at the Indigenous participants in the pilot study. We also saw similar results for a variety of trace elements, such as barium, aluminum, strontium, and manganese. So that initial pilot study allowed us to observe higher levels of some chemicals potentially associated with UNG in pregnant women from Northeastern British Columbia. And we know that these compounds can transfer through the placenta and reach the fetus. So in a larger biomonitoring study, we want to confirm or disconfirm the results from that initial pilot study. Moreover, because endocrine disrupting activities have been observed for a variety of chemicals associated with UNG, we aim to investigate the endocrine disrupting activities of low levels of UNG chemicals that represent, maybe more accurately, the real exposure levels in the fetus. So this leads me to present you some results from the exposures in the Peace River Valley or Xperva study. So this study is funded by the Canadian Institutes of Health Research. It's a very large research team, but I want to acknowledge the work of my co-PI, Marc-André Bernard, and we also put in place an oversight committee whose mission is to evaluate all the aspects of the research project prior to their use. Again, it's a community-based research project, so we work with the same community partners. So the recruitment was conducted in 2019. It involved the participation of 3 medical clinics and 1 midwifery clinic, as well as the community partners who advertised the project to their members. We recruited 85 pregnant women, which represents approximately 33% of the pregnant women in the region at that specific time. It's not a very populated region, as you can see. And another great recruitment outcome is that 90% of the pregnant women that we met decided to participate in this study. So Xperva is a very large study, not in terms of number of participants, but in terms of different analysis. So I won't go over everything, but for the exposure assessment objective, we use environmental monitoring and biomonitoring. So we measured VOCs and trace elements in tap water and indoor air, and we also measured the same chemicals in urine, hair, and nails. We also estimate the UNG activity around the participants' residence. And to do that, we calculate the density of wells around the participants' residence, so just the number of wells, using different buffer zones around the participants' home. We also calculated 4 metrics that consider the number of wells and also their distances to the residence. We did 1 metric accounting for all the wells in the province and 3 other metrics using different buffer zones. Those metrics are called IDW in the rest of the presentation. So in Xperva, the median age of the participants was 29 years old. 20% of them identified as Indigenous. The median gestational age was 23 weeks pregnant. The majority of the participants have had previous pregnancies. Most of them were not smoking during their pregnancy. 23% of them were exposed to secondhand smoke at some point during that pregnancy. 22% of them had an attached garage, and roughly half had city water as their main source of residential tap water. So smoking, exposure to secondhand smoke, having an attached garage, the type of water, those are all contribute to the levels of VOCs we may measure in indoor air or tap water samples of the participants. So that's why we ask those questions in the questionnaire. In total, we identify around 29,000 oil and gas wells in the region at the time of the Xperva study recruitment. I thought it would be interesting to show you the maps that we generate in order to calculate the well density and the proximity metrics. So the first image here on the left represents all the wells that have been drilled so far in Northeastern British Columbia in green. In the second map, you see that the wells are now in pale green, and in purple, you can see where people are living in that region. And here, this is just a close-up of that map, so you can appreciate how close the wells can be to residences. It's important to note, however, that not all those wells are active. Some may be abandoned, decommissioned, or in different phases of production. So we measured a lot of different volatile organic compounds in indoor air. I will go briefly over that. So in this graph here, you can see multiple levels of VOCs. In blue, this is what we measured in Xperva, and in orange yellow, this is what is measured in the general Canadian population. So for 27 out of the 40 VOCs we measured, the median levels in the Xperva study participants were between 1.3 and 3 times higher than the national median levels. And in particular, for the VOCs that you now see identified by red boxes, the proportion of participants from the Xperva study with indoor air levels of those VOCs over the 95th percentile reported in the general Canadian population ranged between 10 and 28%. We then looked at those specific VOCs that had higher levels in Xperva compared to the rest of the general Canadian population. And we looked at their association with well density and proximity metrics. So here, what you can see, I won't go over the methodological details unless someone's interested maybe in the Q&A, but here you can see results of what we call multiple linear regression models between metrics of density and proximity of the wells and different VOCs in indoor air. So briefly, what we see is that there is a significant associations between the number of wells and some IDW metrics and the levels of acetone and chloroform in indoor air. It's also important to note that Indigenous status contributed significantly to those models and that levels of those particular VOCs were higher in Indigenous participants' residence. We also conducted sensitivity analysis to account for other sources of exposure to VOCs, and these additional statistical analysis still supported the associations between UNG wells density and proximity and levels of certain VOCs indoor. So to conclude, we have some epidemiological and toxicological literature that suggests risk for poorer birth outcomes and maternal outcomes, but we still face an important lack of data on exposure assessment and also potential mechanisms of toxicity. In Xperva, we have some preliminary evidence of higher exposure levels to VOCs compared to the general Canadian population. And we also have preliminary evidence that the density and proximity of natural gas wells may contribute to the residential levels of some VOCs. I just want to acknowledge all the members of the team. They're all coming one by one, all right, for their work and their help analyzing all that data. And I just want to conclude by saying that Xperva is ongoing. We hope to have more results in the coming months. We're still actively working with our community partners to develop those research projects in partnership with them and to assess the impacts of those pressures on health by combining information from different levels of biological organization. Thank you for your attention. Happy to answer questions.